The development of the central nervous system is a very relevant subject for those of us clinicians who deal with patients every day. Not only with those patients who have diseases related to a congenital disorder, but basically with any pathology that affects the central nervous system of any human being. Understanding the development of the central nervous system is akin to understanding the history of the nation in order to interpret the current events. Would be preposterous if I would pretend to cover the vastness of the subject in these lectures or in many more lectures, because certainly that goes beyond my intellectual ability. But I would like to share with you some paradigms that are leading researchers today, and also by explaining or sketching some issues regarding the development of the central nervous system, it will arise your curiosity, and you will dwell in depth in the issues that most interest you. The understanding of the construction of the central nervous system can be itemized in 3 points. One is the creation of a shape, the origin and development of morphological characteristics. Then some of those elements created early in the development will migrate to other positions. Typical example are the neurons. And the metamorphosis, which is the continuous shaping and reshaping of the central nervous system even after intrauterine life. The classical example to this issue are, of course, plasticity after any form of brain injury and the modifications imposed on the brain by learning. The beginning of everything and the core plan or the blueprint of the brain is already present in the placote. This is a sketch that I borrowed from the book of Dr. Swanson, Brain Architecture, a book that I strongly recommend to all of you who are interested in the study of the brain. And in this the sketch, what tells us is that every one of the important anatomical structures that will have the central nervous system are prepared to be in the original placote. What do we know of this at the placote? There was one time in which we were a two-layer embryo, and in those two layers, the ectoderm and the endoderm, or the endoblast and the hypoblast, started a conversation. So this it is called neural plate, but of course we understand this is ectoderm, which will be later transferred into the neural plate, and this is the hypoblast or the endoderm, a layer that will later disappear in its current characteristics. But between those two layers, a dialogue begins that helps the transformation of the superior layer influenced by the inferior layer in order to prepare the ectoderm to become the neural plate and prepare the neural plate to become the blueprint for the whole central nervous system. So if we have in yellow here the hypoblast and the epiblast or endoderm and ectoderm, we'll see that FGF, induced by FGF from the hypoblast, the epiblast expresses ERNI and SOX3. Those two structures prepare the ectoderm or the epiblast to begin the transformation of the tissue into a tissue that will be able to become central nervous system. Meanwhile, Cerberus is inhibiting the primitive node or the node of Hansen. Then, when the inhibition of Cerberus stops, the primitive state happens induced by the nodule of Hansen. Besides the precise details, whether this is Cerberus on the primitive node or this is FGF on ERNI or at the SOX3, I am sure that some elements of this sketch will be defined and redefined in the years to come. But the concept here is a dialogue of preparation between one layer and the other layer. So what we have is an epiblast, and slowly we have, after the inhibition of Cerberus, the primitive node and the striate start creating a shape in the center of the epiblast. The primitive node or node of Hansen is a very interesting structure because if transplanted from one experimental animal to another experimental animal, the host will have two central nervous systems. So the driving force, the propelling engine for the creation of the central nervous system is the primitive node. This is the oropharyngeal membrane, a structure that will later disappear. So when we were 1 millimeter in shape, when the day 16 after conception, we already had the core of the developing of our central nervous system. This primitive node and the striate will induce the migration of cells between the epiblast and the hypoblast to form the mesoderm, and that mesoderm later will form the notochord. The central mesoderm will be the notochord, and the notochord will continue the induction of the central nervous system over the ectoderm on top. So to make things clearer than this animation, if possible, we have a primitive node, the node of Hansen, primitive striate, and 3 layers, I'm sorry. Now we have a conversation between the epiblast and the hypoblast, between the hypoblast and the epiblast has been interrupted by the presence of the mesoderm. And the mesoderm, with a precaudal mesoderm, starts conversing with the epiblast and continues to prepare it for the formation of the central nervous system. The BMPs, the sonic hedgehog, nodal, WNT antagonist, all of them are elements that for which you will find abundant literature in PubMed or in your textbooks. But remember just for the moment that they are there and they are continuing to prepare the epiblast to become a central nervous system. An important set of genes, if we have to say the 3 more important set of genes, pair of genes or pair of molecules that work in the for the developing of the brain are the bone morphogenic proteins, the sonid hedgehog, and the HORS gene, of which I will talk later. But at this stage, as the HORS gene is dependent on the concentrations of retinoic acid, at this stage also the node is preparing the terrain for the retinoic acid to be abundant to allow the HORS gene to work on the developing of the central nervous system. The interesting point is that, or the fascinating point, that they, for years it was believed that the ectoderm was basically a tissue prepared to be skin and needed to be transformed into a tissue, some portions of tissue to become central nervous system. Well, apparently the thing is upside down. The neural induction is the default pathway for ectodermal cells. Ectodermal cells are supposed to become a huge nervous system. In order to have skin and epidermal tissue, we need to block that preference of the ectoderm, and the bone morphogenic proteins are the ones who block or are the epidermal inducers. They do not want the ectoderm to become central nervous system. They want the ectoderm to become skin. But then when a series of neural inducers, not CHRD and FST, appear, they block BM4, and as a consequence we have a neural differentiation. So the famous epiblast, or the ectoderm, was supposed to be a huge brain, and it's not a huge brain, it's just limited to the central axis because of the work of BMP4. So by day 16, when we were 1 millimeter in shape, begins a process in which the cells at each side of the group elongate. They become taller, and they create a groove, and they converge, and we are seeing in this animation that by day 22 the typical shape of the embryology textbooks, that when we see the developing of the brain, is appearing. The concept to be remembered from this animation is that the cell elongation is an elongation of the cells. It's not that there are many layers of cells, it's a single layer of cells, and that the orientations of the cells is such that allows the lumping of the sides of the groove to create like the mountain that will eventually fold. But then we have to have the furrowing and unfolding, in order to create the appearance and the shape of a tube. The cells from the future neural tube experience transformation at 2 points: at the medial hinge above the notochord, if you remember, was the area induced by the mesoderm, and the lateral hinges. So the neuroectodermal cells have a process of wedging. Actin binding proteins change the shape of the cells. Unfolding folding is mediated by lateral non-ectodermal tissue. This final process of furrowing, folding, and closing, if you remember, we were one big slab of tissue with a differentiated tissue in the center, but basically the neuroectodermal was adhered to the regular ectoderm, happens through a process of kinking first, that this is the neural plate, this is the regular ectoderm, the epidermal ectoderm, in which the cells of these sides of the neural plate are taller than the cells on the regular ectoderm. That forces the creation of a process called the delamination, and finally, by having the hinging, the dorsal hinging plate, the two tissues separate. So what happens? What's the clinical problem related to that? And of course, it has jumped to your minds already that the neural tube's defects, in all its many variations, will occur if this process of furrowing and folding fails. We all know, and it's there on the screen, that folic acid is a very important participant in this process. It is not known yet how, but it is known that if the process of wedging and working of the cells, or transformation of the cells at the level of the hinges, is an actin or a protein-based process, and any abnormality in the metabolism of the proteins causing altered protein function will cause a neural tube defects. It is known that is something secondary to altered folate metabolism. This important paradigm in the understanding of congenital diseases happened, or was presumed, after a very smart clinical observation of an obstetrician who observed that neural tube defects was more common, or were more common, in patients who had megaloblastic anemia. So he presumed that the alteration of the metabolism of the vitamin B12 could be responsible for this increased incidence of neural tube defects. The paper that is quoted at the end, at the bottom of this slide, says it is called "Not everything is understood yet." I mean, although we tend to believe that by giving folic acid to every woman in the age of conception will erase completely the incidence of neural tube defect, it is clearly no, because there are genetical factors, and the environmental factors maybe are wider than just the folate metabolism. But nonetheless, if we look at the incidence of neural tube defects according to regions of the world, we'll see that developing nations have a lower incidence than clearly non-developed areas of the world. Mexico, who belongs to North America, has a higher incidence than United States and Canada. Whether this is because of the supplementation of food with folic acid, or whether this is because the prenatal diagnosis of this condition leads many of the parents to opt for termination of the pregnancy, still has to be determined. But certainly, folic acid prevention plays an immense role, and all those efforts have to be praised. Interestingly, and as I mentioned before, in the many environmental issues that have not been clarified yet, in Guatemala they forced folic acid prevention into the corn, but sometimes the corn for which the flour is obtained and is the staple food of the population in the highlands of Guatemala, where there is a very high incidence of neural tube defects, that corn is infected by a fungi, and the fumonisins of this fungi eventually block the metabolism of the folic acid that has been added to the food. A process of nixtamalization is advised to prevent the developing of these toxins of the fumonisins. The idea of sharing with you this information is to actually tell you that the problem is not just simple as the fact of giving folic acid, and through folic acid assure that the actin binding proteins will work adequately and the furrowing and folding of the central nervous system will occur. The idea that we obtained from the illustration or from the animation 2 or 3 slides ago is that there is a one single starting point for the closing of the central nervous system, and it is not, and that's why we have areas of defects of neural tube that vary in different regions according to where and when this closing defect took place. These issues will be analyzed extensively in the lectures specifically addressing those pathologies. The concept here being that the closure of the neural tube defects does not start like a zip in one point and ends on the other point. It starts at different points. So, at the recap, we had a flat slab of tissue. We transformed the center of that tissue into a tissue specific to become central nervous system later. That tissue was closed, has created a neural tube, and we have below that neural tube a notochord, and we have that neural tube has been closed with epithelium on top. What happens to the future spinal cord? Our old friend, the BMP4, will be the inducer of the dorsal aspect of the cord, which is the sensory motor. So BMP4 induces sensory neurons and interneurons, and the sonic hedgehog related to the notochord induces motor neurons and interneurons. This is done through the activation of a class of progenitor proteins, for which those of you interested will find abundant literature, and is a a great field of investigations for developmental neurobiologists. But the concept as the clinicians again is that the actual tube will be, that the cells within that tube will have identities according to the different gradient concentration of sonid hedgehog for sensory neurons and interneurons, and BMP4 for sensory. I'm sorry, SHH for motor and BMP4 for sensory. And here we come with a third important gene that we had so far. We had the BMP4, which is a set of the proteins, the sonid hedgehog, and the horse genes. This is an evolutionarily highly conserved set of genes, encode transforming fractals from the midbrain and hindbrain, determines the position and function of cells in specific segments of the body, and is regulated by retinoic acid. Remember that the nodule of Hensen and the primitive striate, when withdrawing, when shrinking and disappearing, they were preparing, they were leaving behind a stock of retinoic acid, so the horse gene can fulfill its function. And what the horse gene is doing is, as it says, determines the position and function of cells in specific segments of the body. This is a specific issue of patterning. So in this simple drawing inspired from Dassen et al. In Nature in 2003, so we imagine a schematic spinal cord, brachial, thoracic, and lumbar, and lateral motor column, or the Terry cells, and these cells in here coming from the horse gene 6 as opposed to the horse gene 9 and the horse gene 10. These cells in here will be specifically prepared for working together with the hind limbs, with the upper extremities, as opposed to these cells in here will work only with the lower limb. That specific patterning on our the spinal cord is a responsibility of the horse gene. So we had part of the patterning was coming from the ventral aspect by the sonid hedgehog, part of the patterning from the sensory side, from the dorsal side, was coming from the BM4, and that tells us which one were sensory and motor neurons. But now we needed to say, okay, this set of motor neurons will be specifically allocated to the upper extremity as opposed to that other set of cells allocated to the lower extremity. And with this, we're more or less finished with this overview of the primary neurulation. Primary neurulation was then the process depending on the ectoderm, a process in which a tissue is differentiated and affected in its differentiation by the constant dialogue between the epiblast and the hypoblast and the mesoderm and the epiblast. There is almost like a footnote in development, which is the secondary neurulation, or the second neurulation. And here we see the formed neural tube, the bent embryo, and the second neurulation, basically what is a process of canalization. Caudal mass vacuoles coalesce and contact the central canal and create the ventriculus terminalis and the filum terminalis. It takes place 7 weeks after birth, 7 to 7 weeks after birth. The disorders of the second neurulation will develop according at what time that process is altered. So if you have a process of secondary neurulation altered at the very beginning, at the 7th week, the patient may develop a myelocystocele, then either a diastematomyelia, meningolipo, meningocele, lipoma, teratoma, dermal sinus, and finally the easy to treat and almost benign but not innocuous thickened filum terminalis. So all of them are different processes of the secondary neurulation, which is basically, as we saw in the very short and simple animation, is a process of coalescence of cells that vacuolize and join themselves with a neural tube defect, with a neural tube. They want to be part of the central nervous system. So we have also we are focused on the brain, on the spinal cord, but things are also happening on top, and the closed tube has created these vesicles. Those vesicles are bending, are taking a different shape, coming closer to the typical image that we have of the brain and of the spinal cord. The changes that we expressed on the spinal cord, in which the different molecules modify the tissue over which they exert their function, is called patterning. And patterning is the key word for understanding the current orientation of the central nervous system, of the development of the central nervous system. People assumes and understands that there is a specific regions that will fulfill only a specific function. And remember, as we said, on the spinal cord, Hodge gene, SHH, and BMP4. So various signaling pathways direct specific regions of the neuroectodermal to adopt forebrain, midbrain, hindbrain, or spinal cord fates. And this illustration, this is a schematic illustration from Vieira et al. From International Journal of Developmental Biology of 2010, shows precisely that, and is a modified phenomenon, or a modified or a more sophisticated presentation than the first or the second image I show you of the neural plate with all its fate already represented. The complexity of molecules involved in this pattern is clearly expressed in here. So that the pattern in the hindbrain begins immediately after tube closure, is manifested by development of rhombomers, which are transient structures, and cells within each rhombomer are segregated from cells from the adjacent rhombomers. Each rhombomer cell interacts with each cell within its boundaries and does not affect the boundaries of the next rhombomer. So if we look at the hindbrain, we'll see that there are 8 rhombomers, and the Hodge gene controls the positional value of the cell in each rhombomer and is regulated in dose-dependent fashion by the retinoic acid. Excess of retinoic acid may cause a series of congenital malformations, similarly to lack of retinoic acid. The rhombomers as such are the proliferation in the cells occurs in waves, being the first wave of rhombomers are the 2, 4, 6, 8, and the second wave the 1, 3, 5, 7. And as you can imagine, the different cranial nerves will be allocated to different rhombomers. Again, the interesting concept in here is this patterning phenomenon, something that was not that prevalent or clear for those of us who studied neuroembryology in medical school decades ago, and is more prevalent and clear for current medical students. In this image of an embryo, you can see the rhombomers here in the hindbrain. But the hindbrain, the phenomenon of the hindbrain, do not affect the midbrain. The midbrain is the only area of the embryo that will not have any segments. We'll have segments in the telencephalon. We have segments, the rhombomers on the hindbrain, but we don't have segments in the midbrain. The isthmus is a critical region that, through the expression of different genes, creates the boundary and says up to here is the hindbrain, from here is at a different story. Our old friend, the sonic hedgehog, and we'll talk about him in the coming images. I don't know why I say him. We'll talk about it in the coming images. As you remember, it was expressed by the notochord, was anterior, then by the precaudal mesoderm, and the sonic hedgehog also is expressed in a peculiar area of importance for the brain, which is the zona limitans intrathalamic. The zona limitans intrathalamic is a well-divided boundary between the three regions of the prosencephalon, P2 and 3 and P1. Overexpression of the sonic hedgehog results in expansion of the posterior boundary of the prosencephalon and underexpression in the anterior boundary, as you can imagine. And this is the telencephalic vesicle in blue. The central story of the lecture made so far is the regionalization of the areas of the brain, each one of them under the control of a specific set of genes for which the environment has to be prepared. The name of those genes, the details of those genes is something that can be updated through the literature, but that's the concept I want you to actually master. I mentioned that there are 3 prosomers in the prosencephalon, and again we go back to the image of the study from Vieira et al. That it clearly shows that there is a patterning phenomenon, and I'm already repeating it myself, and I presume that some of you will say, okay, I got it. And the final example, and the final nailing or the final nail in the idea or in the concept of the importance of the sonic hedgehog as a morphomer came with this set of experiments by Brito et al. In Nature, published in Nature Neurosciences, in which they see that if they play with the sonic hedgehog influence on the telencephalon and the diencephalon, the telencephalic vesicles, which are normal here, do not develop normally, and a series of aberrations occur when the sonic hedgehog is impaired. The sonic hedgehog is a very interesting gene. Is its developers have earned the Nobel Prize of Medicine and Physiology for their work, and there are basically three hedgehogs: the sonid, the Indian, and the desert hedgehog, and they have a very curious and interesting story behind them. But for the purpose of our lecture, it's worth to emphasize that the synthesis of sonid hedgehog is cholesterol dependent, and the cholesterol synthesis is disrupted in case of maternal diabetes, excessive alcohol consumption, fetal alcohol syndrome, cyclopamine, which is an anti-neoplastic drug, and other risk factors like toxoplasmosis, syphilis, rubella, you name it. The cyclopamine phenomenon is an interesting phenomenon because also leads to one of my favorite the teaching points, which is through clinical observation, deep insight can be obtained on the understanding of normal and abnormal physiology. In the 1950s, it was observed that in a farm in Iowa, I think, a set of lambs were born with one eye. The curious functionary of the Department of Agriculture decided to investigate in the issue, and he found that the sheep were grazing a particular corn lily. He analyzed the structure of that corn lily, and he found a factor that affected the synthesis of sonid hedgehog. This eventually that you have a drug or a product that can affect the morphogenesis led people to say, okay, this is part of an issue relevant to understanding of the brain, until somebody else says, well, if we can affect the growth of normal cells, perhaps we can also affect the growth of cancer cells. And cyclopamine is used for the treatment of medulloblastomas, among other cancers, but also is still an evolving field of work to see how we can alter the metabolism of sonid hedgehog in cancer cells, of course, in the post-uterine life. The development of the prosencephalon, and this is related to the sonid hedgehog, this is related to the zona limitans intermedia, has a peak time of 2 to 3 months, and basically can be divided in three very well set stages: a stage of prosencephalid formation, a stage of prosencephalid cleavage, and a midline prosencephalid development. The details of those the stages are abundantly explained and expanded in many basic embryology textbooks. And for those clinically oriented, I strongly recommend the book I just mentioned above in one of the slides, the book by Dr. Joseph Volpi on neurology of the newborn. But going back to the sonid hedgehog and to the clinical factors, so we had that the sonid hedgehog is a morphogenetic gene, a gene that creates shapes. Alteration of the sonid hedgehog will impair the development of the prosencephalon. And those pathologies that have impaired development of the prosencephalons, according to which stage they do happen, can be aprosencephaly, atelencephaly, holoprosencephaly, and the agenesis of the midline structures. Holoprosencephaly, which is a disease not that infrequent because it's in 1 in 250 conceptions, but fortunately, shall I say, only 1 in 13,000 live births, is a disease that has attracted the interest of investigators because of the window that it can open towards the understanding of the developing of the brain. Even the most basic-oriented textbooks on neurodevelopment do have a chapter on holoprosencephaly. For us clinicians, holoprosencephaly represents a challenge because we sometimes have to shunt the enlarged ventricles if they do cause intracranial pressure, but most of the time the pediatric neurosurgeon is the person to whom the families come for an explanation and understanding of the condition that affects their children. And there are 3 types of holoprosencephaly: allobar, semilobar, and lobar. The lobar is only the anterior and inferior portion of the frontal lobes is fused, as opposed to the allobar, which is a complete absence of interhemispheric fissure, folds, corpus callosum, and septum pellucidum. Everything is a big cavity. And the semilobar, which you have a separated occipital and parietal lobes, but have fused frontal lobes. The severity of the clinical manifestations, of course, varies according to the severity of the malformation. Many patients with lobar prosencephaly have basically a normal life. What is interesting also is that the face or the facial structures of the patient, some of them with cleft palate, some of them with hypertelorism or broad nasal ridge, will go in parallel with the severity of the damage to the central nervous system. Both things are understood if we remember again that the sonid hedgehog is basically a morphogenic gene that alters shapes and configurations, and the prosencephalic development is linked through the SHH through the development of the facial structures. So we can say that we have reached now a moment of, we have finished this part in which the shapes have been created. We had a single slab. That slab was modified by the conversation with adjacent layers, BMP4, SHH, horse genes, creation of rhombomers. A series of other genes create different areas and different structures, and of course the overall shape of the embryo has been changing. The telencephalic vesicles have become larger and bending, and the cerebellum has been formed, and of course the neural crest and all the adjacent structures have happened. So we have finished with this rapid overview of what is the shaping of the central nervous system. The 2 more important pathologies were the neural tube defects with its different variations and the holoprosencephaly with its different variations. But now we had the creations of the cells. So in the periventricular area, this will be the lateral ventricle, in the ventricular zone geminal matrix, this is the animation of the phenomenon that we see in here, that the cells sort of bounce back and forth between the apical and basal part of the ventricular zone or geminal matrix. They divide. One mother cell and one of the sister cells will eventually migrate and travel towards the cerebral cortex, while the other cell will remain in the ventricular zone to continue with that cycle. In the S phase, which will come in soon, we have the second cell ready to go out and following the radial glia, again, as I said, to go to the cortex. The drawing here is from Fujita from 1963, but has been reproduced intensely and widely in almost every textbook of clinical neuroanatomy even. So what we have seen here, we have seen that we have a group of primitive cells in the germinal matrix or in the subventricular zone, and this group of primitive cells have a potential for developing the neurons and the radial glia. Radial glia, although it's called glia, is another form of embryonic neural progenitor because they undergo symmetric division, giving rise to two radial glia or to two neurons. The process is complex per se, but bear in mind it's controlled by the Hess genes and by the Notch proteins. Mother cells, radial glia, neuron, oligodendrocyte, astrocytes, or ependymal cells. The VHLAH group of genes determine also whether the primitive neuron will grow that will be an astrocyte or will be a neuron. The key issue to remember here, besides the genetic influence, VHLH, Hess or Notch, on the final fate of what the cell will be, is that we have a progenitor, that is the periventricular zone, and another progenitor, which is the so-called radial glia. Calling it glia gives you the idea of that it is a matter of it's a scaffolding, just like a secondary material ready there to help the migration of neurons, when actually it's not. The radial glia will give origin to the neurons and will give origin also to other cells, this time glial cells. So we have to look at the radial glia, as I said, as a embryonic neuronal progenitor. But now, what is another source of cell genesis? Is very interesting, is this one, is the ganglionic eminence. So far, we're looking at the ventricular zone. That's what we show. But in the ganglionic eminence, which are three ganglionic eminences, medial, lateral, and caudal, the median and lateral will form later the basal ganglia, and the caudal ganglionic eminence will be part of the limbic system. Those cells generate inhibitory GABAergic neurons. So we have two areas of origin of cells in the mammalian brain. The ventricular zone, the most common process, the one that we all know about, and the forgotten process, on the ganglionic eminence. And as we have two different zones of source of cells, we also have two ways of cell migration. From the subventricular zone and the ventricular zone, we have the radial glia, and the cells migrate climb along the glia to the different layers of the cortex. Perhaps it's familiar to you, this is the concept that the last wave of neurons to migrate will go over all the other layers and will go to layer 1. And the first layer of the first wave of neurons that migrate actually will be in the deeper layer. So the last to arrive go to the top. So that's the radial migration. But there is a transverse migration coming from the ganglionic eminence. And the progenitor cells usually migrate through the marginal zone or the intermediate zone, and then eventually may even go, those interneurons, those GABAergic interneurons, take a small jump through the radial glia. But there are two ways of cell migration. There are two sources of cell production, but the production of cells in both areas are regulated by the same group of molecules, the His, the Notch, and the VLHAL. So fine, the neurons arrive to the cortex, and how they do what they have to do. And this is an issue that has not been completely settled so far, which is when the neuron that will be a motor or sensory neuron determines its fate, when the neuron that the visual and auditory cortex determines its fate. Some experiments have been working around, and a couple of the structures have been identified that different regulation of this the transcription factors EMH2 and PATS6 will create either visual, motor, or sensory neurons in the experimental animal. So the EMH transcription factor is denser in the posterior and medial. The PATS is denser in the lateral and anterior. So if you have a mutant, very little of EMH2, you will have a very small visual cortex, right? You will not have the posterior portion. The visual cortex of that experimental animal will be reduced, and the motor and sensory will be expanded. Whereas if you inhibit or mutate the expression of PAT6, the animal will have a very little motor cortex and very big visual cortex. Again, this is a process not totally defined, but serves this elegant experiment to arise your awareness of those fundamental questions that are still not fully explained, but that are a very interesting question. Is what decides the fate, how the neuron becomes, or the area of the brain becomes the area that it is. So in this the core phenomenon of the cell cycle and the migration is to give you a phenomenon that is a dynamic process. It's not just cells from point A to the point B in an order fashion. It's an integrated process in which you can see in this sketch that the ventricular zone or the intra or the subventricular zone become narrower, the cortex becomes wider, and the intermediate areas change as well accordingly. Similarly, on this slide, in this illustration, I'm sorry that more than pay attention to the meaning of that the colors gives you a sense of patterning. You see this blue from the intraventricular zone, so from the subventricular zone, then expansion, then decrease of the subventricular zone. There is an intermediate zone that also almost disappears, whereas there is a cortex that from being barely seen or barely represented finally reaches its complete and final shape. This is seen in here in the animal, on a pathological preparation of a human fetus, and the MRI of the human fetus as well, that you see in the beginning you have a ventricular zone, an intermediate zone, and a cortical plate, but then you have a ventricular zone, intermediate zone, a subplate neuron, and a cortical plate. So a dynamic phenomenon of migration, of active movement of cells as the embryo is growing inside of the mother. The subplate neurons, I bring you back again in here. We saw them in here, and the subplate neurons will be expressed much clearly in the same illustration here. Are an interesting set of neurons. They arrive before the cortical plate, linked between afferents and cortical neurons, provide extra guidance for actions from the cortex, and 90% undergo apoptosis postnatally. And the disorders that we will see, disorders of neuronal proliferation, microencephaly, macroencephaly, and disorders of neuronal migration, schizencephaly, lysencephaly, polymicrogyria, heterotopia, focal cerebrocortical degenesis, phenomena that will be thoroughly explained later when the lectures on epilepsy and epilepsy surgery are presented. But that's, you have the, hopefully I was clear enough for provide you with someone with understanding of the core seat idea why those diseases happen. So we are coming to the final process of the development of the brain, and for the 9 months we have the fully developed central nervous system that has been so far a matter of migration and morphogenesis. The morphogenesis was determined or predetermined by the interaction of different molecules. The migration is predetermined by the interactions of a different set of molecules, and the reason that different cells have their personality has not been clarified yet. But we go into the extrauterine life, and there are some areas that still have a clinical relevance. Dr. Quiñones Hinojosa from Johns Hopkins has done extensive work on this portion because of the following. He has paid attention to the subventricular zone. If you remember, the subventricular zone was very prominent in the beginning when the development of the cell cycle, and then as the cortex is already taking its final shape, the subventricular zone became a tiny layer of tissue, usually relegated to the periventricular area of the forebrain and some areas of the hippocampus. But still, in that subventricular zone, that in the human has 4 layers, it has a layer of ependymal layer, has a layer of a gap without cells, with astrocyte, and then comes with the brain parenchyma. In that area, we have the stem cells that are so invoked nowadays with regards to their therapeutic potential. And also we have the supposed potential for neurogenesis. The question is, do adults or adult brain generate new neurons, or all the neurons were already created during the intrauterine life? The jury is still out about that, but the core center of the stem cells is the subventricular zone, and the core center of after delivery neurogenesis is also in the subventricular zone. This has an importance, a clinical importance to begin with, because of the subventricular zone hemorrhage. The other name, or the old name of the subventricular zone, if you remember, was germinal matrix, because it is the matrix that gives origin to the neurons. The subventricular zone is located, basically, the remnants of the subventricular zone are in the vicinity of the head of the caudate or in the forebrain periventricular area. You don't have a subventricular zone in the parietal or occipital areas of the lateral ventricles. So it is limited because of the peculiar arterial-venous interaction of that area. That region is prone to bleeding or ischemia. The bleeding and the ischemia that we see in our patients in the neonatal unit will impair the development of new neurons, if there are still new neurons to be developed, or will also impair the potential plasticity that can be induced from the subventricular zone. This plasticity may not be only related to neurons, but may be also related to glial cells. It is hard to know, but not surprisingly, considering the importance of the subventricular zone, we see that premature infants with periventricular leukomalacia do have a decreased cerebral cortical gray matter volume, do have impaired IQ, and this is from an experiment in rats that different time of ischemia without being fatal, without being causing the bleeding, depletes the population of the subventricular zone. So, and those experimental images on top are from postnatal animals, of course, and this is the phenomenon that we see of impaired development, although in premature babies, but sufficiently advanced in their intrauterine life as to be able to survive through the neonatal intensive care unit care. So the answer is not completely clear about what's the role of the subventricular zone in neurogenesis, but certainly a damage to the subventricular zone impairs the intellectual development of the child. And going back to the neural stem cells, which are in the subventricular zone, so the stem cells are a minor subset of cells which have a cell renewal and are multipotential, can give origin to different type of tissues. The stem cell for the whole body is the embryonic stem cell, and then there are organ-specific stem cells. So you will have a neuronal stem cells, you will have an intermediate progenitor, and that stem cell will give origin to different cells according to what is needed. That neuronal stem cell is in the subventricular zone. how can we profit from that pocket of potential developed neurons or oligodendrocytes or astrocytes is not clear yet. Dr. Quiñones Hinojosa has been working, and also this is from our group here, that you can have a pre-malignant stem cell still in the subventricular zone that will become a cancer stem cell and differentiated cancer stem cells as well. So the neuronal stem cell and the brain tumor stem cell do share a series of pathways, regulatory pathways, cell markers, affinity for basement membranes, migratory capabilities. And again, that brings the famous question of, do we have a neuronal stem cell or a cancer stem cell that can be early identified, or treatment of cancer, of brain cancers, can be elucidated through the understanding of the biology of the brain tumor stem cells. There are tumors that grow in the vicinity of the ventricles, gives you the idea or the geographical or anatomical impression that they come by association. You have the subventricular cancer stem cells, and the glioma arises near to the subventricular area. But as we all know, many of the tumors grow far away from the lateral ventricles. Whether this has to be with the migratory capabilities of the brain tumor stem cells, we don't know yet. And the other dynamic process that happens endlessly after life, some people say after until the first 2 years, but perhaps it's much more than that, is the process of myelination. Myelination is what increases the speed of conduction of the neural impulses on the central nervous system. Couple of basic concepts: begins in the human in the spinal cord, later goes to the cerebral cortex, and sensory and motor system becomes myelinated before the cognitive processes. And in the brain, the myelination has also an order. First, the posterior frontoparietal occipital sides, later the temporal sides, and finally the anterior frontal sides. This process also is related to a very interesting phenomenon, even from very recent data, or very recently published data, that shows you that a 12-year-old will have not as much, will have more gray matter than a 20 years old. So we'll have a decrease on our gray matter as we age. That decrease could be because of pruning of the neurons, but also because of increase of the myelination. So this active conversation between the myelination and the pruning of the neurons may be responsible for this constant shaping and reshaping of our brains as we learn, as we acquire experience, and as we become more accomplished in our passions and our profession. Good if our passion coincides with our profession. That's a fantastic life. And this idea of a constant changing and pruning and myelination, and the interaction between a brain and its environment, because as you can guess, myelination depends on food, myelination depends on the thyroid hormones, myelination depends on training and exposure to motor activities. The neuronal pruning is not just an automatic pruning. We will modify this cerebral cortex because you are 15 or 20 years old, we will modify the cerebral cortex according to the stimulus that you have received, according to the environment where you live. So this is not surprising if you look at these compared maps in which areas of undernourishment, and you can identify the geographical regions easily, is a living contrast with the areas of where there is abundance of science research. And although this may be also, of course, it is related to the availability of funds for developing universities, centers, and funding basic research. But also has to do with the developing of the brain as a biological structure impacted by the environment. So in Proceeding of National Academy of Science in 2009, there was this very interesting graph which tells you that the duration of childhood poverty and the working memory in young adults is inversely related. Longer childhood poverty, less working memory in young adults. The anatomical justification for this was shown in 1999 by a group from Mexico City, taking advantage of almost a obligatory regulation in the hospital where I had the privilege to work many years ago, Hospital Infantil de Mexico, where almost every child who dies in the hospital has to have an autopsy, or the autopsy is offered, and many patients do have autopsies. So because of the importance of the hospital in a central hospital in the country with such social disparities as they exist in Mexico, they were able to compare brains of children who died for other reasons, I mean no neurological reasons, but children who were undernourished and children who were normally nourished, and the distribution of the dendrites and the apical growth from the axons was significant when compare normal infants with malnourished infants. So the anatomical structure that allows the developing of the normal intelligence is seriously affected, not in the intrauterine life, after the child is born, because the same active mechanism of growing and developing a brain are not stopped and altered immediately after birth. So hopefully this small lecture has give you some understanding of the paradigms that are currently running the study of the development of the central nervous system, made you familiar with some names: Sonic Hedgehog, BMP4, Hodge genes, Notch, Hess. But for those of you who really want to expand and go one or two at a step beyond, there are two highly detailed books, Developmental Neurobiology, both of them, called one edited by Greg Lemke and another one edited by Rouse and Jacobson. And on the right, two other books who are more easy to read: The Fundamentals of Brain Development, which is a very good book. But if you have to choose one of those four books to read independently of your interest or not in the subject of this lecture, I strongly recommend Brain Architecture by Dr. Swanson. And on that note, thank you very much, and we'll be expanding on the clinical importance of those issues in future lectures, and we will be coming back to this very preliminary introduction to the development of the central nervous system once we start working or start developing the clinical cases that are the ones that truly interest us.